So if we go back to our picture here of, we're frozen, sorry, of what's happening, this is a very good diagram in that it's really showing a summary of what is happening. We're getting light coming in. We're still talking about light reactions. We still have not talked about Calvin cycle. And we will probably touch on that today. Light's coming in along with, I'm going to talk about, whoa, that's too big. We're going to talk about today, let's see, is that better? Yeah, that's much better. We're going to talk about water today and its role a little bit more. And this is all happening in our thylakoid membranes. This is our thylakoid here. And we're making ATP and we're making NADPH out of it. The thing that's being released that's important to us is oxygen. In that second part that we'll start talking about today, the Calvin cycle, that's where we're going to use these two things, the ATP and the NADPH. Basically, all we've done in the light cycle is we've taken our sunlight and we've turned it into a chemical form of energy that our body can store. Okay, ATP and NADPH can be stored. And we're going to store those in the form of glucose through the Calvin cycle. Okay. So I want to go back to this picture that we talked about yesterday here. And kind of do a little more talking about what's going on here. This is all happening in our thylakoid membrane. Okay. These purple things are called photosystems. They are proteins. Embedded in those proteins are chlorophyll molecules, chlorophyll pigments. And those chlorophyll pigments are going to be responsible for capturing light energy. That energy is not easily contained, and it tends to bounce around from one carrier to another, or I should say, in this case, from one pigment molecule to another. Hopefully I can still read that. These are, make that bigger too. These are chlorophyll B pigments. They don't really have the capacity to hold on to this light energy for very long. So they take that photon and they pass it to another one, and they pass it to another one. And it's kind of a random process. They keep passing it on from one chlorophyll B to another chlorophyll B to another chlorophyll B until it gets to a chlorophyll A. That's a chlorophyll A molecule. Chlorophyll A is a very, very strong electron acceptor. It draws that photon of light. And it also is going to be responsible for the other part of this, where those electrons came from. Those electrons that are going to receive that photon of light are coming from water. Okay? Water is going to give up its electrons. It's going to give up its protons. And when it gives up its electrons, that will be what receives the photon of light. Okay? The photon of light is like your hand hitting that ball on the tether. Okay? The ball in the tether is the electron. Hit it, and now that electron is spinning in a higher orbital. And so that's why we show it as being excited, because it now has more energy than it had before. Those electrons came from the splitting of water. They came from the water molecule. The photon gets bounced around from B to B to B until it gets to an A. The A takes the water, splits it into an electron and protons, and then that electron gets passed from carrier to carrier because now it's excited, it's energized. It's like that ball on a string. It's got a lot of momentum behind it. And as it does that, it's going to be passed from carrier to carrier. And as it does so, it will use a little bit of its energy every time it gets passed from one carrier to another to pump a proton against its concentration gradient. Remember that from cell respiration? Yeah. Okay, let's go to that picture. If I can do this. I'm going to go over it again. Okay. Again, we got a lot going on here. Let's see if I can make this just a little bit bigger. Oh, that's as far as I can go. Okay. This is my thylakoid membrane. Okay. And I have these proteins that have the chlorophyll molecules in them. Light's going to hit that. It's going to hit that B molecule. It's going to get bounced around. And it's going to excite my electron. It's supposed to be my electron. Sorry about that. And it's getting passed from one carrier to the next. Gets passed here to here to here. As it does that, it's not the button I wanted, sorry. As it does that, hydrogen ions are going to get pumped against their concentration gradient like they did in the electron transport electron transport chain in cell respiration. Same thing's happening. We're generating a whole bunch of H pluses, but they don't want to be here. Where do they want to be? They want to be outside. So they're going to flow down their concentration gradient out here. And as they do so, that generates ATP. Okay. So we excited that electron. We liberated it from water. We excited it. And as it was passed from one carrier to the next, it pumped those H pluses inside. They flew. They went back down their concentration gradient. And as they did so, they generated ATP. Yeah, that's that chemiosmosis that we've already been talking about from cell respiration. Yeah.
So is it like the reverse of like oxidative phosphorylation happening? Because aren't they like pumping hydrogen out and then it's like falling back in?
Yeah, that's good. It actually is the same process, but it's in the other direction. You are correct. Yeah. Yeah. And if you don't understand that, don't worry, because there's a slide to come on it. But we're making ATP. That electron's not done, though. Its job isn't done. It's going to go to a second little protein here. What color have I got? Okay. That's my second protein. It's going to get excited again. It's up here and excited again. But now, rather than generating more ATP, it's going to get transferred to a molecule of NADP to become NADPH. What did I say happened to NADPH when it went through cell respiration? Generated a whole bunch of what? ATP. Remember how I said that an NADH molecule will make 3 molecules of ATP? And FADH2 will make 2 molecules? Remember that? Okay. So there's a lot of energy in here. There's a lot of energy in the NADPH. That's what I want you to understand. A lot of energy here. Obviously, there's energy in ATP that we made here. So put my ATP here. We went through all of that struggle so we can take these guys to the Calvin cycle. Because we need to store that energy in a form that's usable. And that form that's usable is sugar or glucose. All right. I'm going to make this bigger. Questions? Mm-hmm. Right, to make ATP. Right. No, not from the electron transport chain. That generates ATP just like cell respiration did. But instead of ripping the electrons away from NAD, we're actually giving them to them. We're reducing it rather than oxidizing it. But it's still a carrier. Mm-hmm. Yeah, it's still carrying it, and it's going to carry it to Calvin to make sugar. So those electrons are high energy, but we don't want to lose. We don't want to spend that energy yet. Tyler.
So I know the first process is like the second one creates ATP, but it goes to the second one to make NADH.
Yep.
So like, how does it go? Like the first one to the second one? Does it just get one energy from?
It gets another photon of light.
Okay. And it's only 2. Like this is.
Yeah, it happens twice. So it gets bounced. It gets hit with 2 photons of light.
And is there like a reason, like why doesn't it just like
oxidize it, breaking down?
Because it also needs those protons to generate. It's C6H12, right? You need those hydrogens. So it's going to use those hydrogens from the NADPH to make the C6H12 part of the glucose. Yeah. So it needs both those things. Yeah. Yes. Are you talking about this one? That's an electron being transferred. So I've got light coming in. Light's going to hit the electron and boost it to a higher energy level. And then it's going to be carried or passed from one molecule to the next. And as it does that, that causes the protons to come inside. Okay? Good. All right, good questions, guys. All right, more questions. Anybody? Hit me.
Where'd the clock go?
The clock is over there. I don't appear to be able to fix it. And I put in a request for a new one. So it's coming back. Here's an analogy, maybe help you with some of this. We got a little worker bee here down at the base. That is like a photon of light. The guy's hitting it. And as he hits that teeter-totter, that electron gets boosted to a higher energy level. Oh, worker bee. Oh, that's too small. And then this worker bee is going to let go of that electron. It's going to roll down this little ramp. As it rolls down that little ramp, that's where we have the wheel. Remember we talked about the water wheel and how that generates the ATP. So we're generating ATP that first time it gets hit with a photon of sunlight. The second time, got another worker bee, hits it again. Whoop, goes up here. Now it's got a lot of it's got a lot of energy, but rather than using that for anything, see how it's just kind of stored here? That's to represent that. Obviously, if this was, instead this was like a bowling ball, and you let that bucket, you tip that bucket, there's a lot of kinetic energy there. There's a lot of force behind there. Same thing with this electron. There's a lot of energy there, and it's all stored in that NADPH, stored in that bucket. That's going to be moved to the next part of photosynthesis, which is the making of sugar. Okay. We did all of this just so we could change light into a usable form of chemical energy, ATP and NADPH. That's all we did. It is crazy.
I think I am.
Frozen here. If you like to see things written rather than visually, you can look at the descriptions here. There's 8 steps. A photon hits a pigment in the light harvesting complex, and that energy gets passed on to the pigment molecules until it gets to they're calling this P680, which is a fancy way of saying chlorophyll A. The excited electron gets transferred to a primary electron acceptor. At the same time, water is going to be split, transferring its electrons. Those are the ones that are going to be energized. One of the things that comes out of this is, and this is a question that comes up often on tests, is where does the oxygen come from that we breathe in photosynthesis? And it's from the water. It's not from the carbon dioxide. A lot of people will say it's from the carbon dioxide that they take in. They actually use that oxygen to produce the glucose. But they don't use the oxygen from the water. The water gets split and the oxygen gets released. Each electron falls through a series of steps in that electron transport chain. And as it does, that creates a proton gradient. And then you get that potential energy stored in the proton gradient to use to make ATP by our wagon wheel. Remember our wagon wheel, or not our wagon wheel, our water wheel. Got our water wheel moving and generating, as it spun, that generated the production of ATP. Same thing is happening here. Electron gets hit by another photon of light. And this time it's going to be passed to NADP positive to become NADPH, so it becomes reduced. Just means that the electron gets a taxi, and the taxi is NADP positive. And then those electrons will be used for the Calvin cycle. All right. Already talked about the analogy. There's some other slides in here that I've taken out that we used to need to know. We don't need to know it now. Chemiosmosis. Chemiosmosis is that generation of the proton gradient that once they come back in or back across the membrane and over that ATP synthase, it generates ATP. Right? Talked about that before. Now you just go to the picture. I think that would be easier for you. So electron transport chain pumps hydrogen ions against their concentration gradient. So they keep pumping them. So there's lots of them here. They're really not happy to be there. And the chemiosmosis part is Then flowing back down, their concentration gradient. Yay. Happy hydrogen ion. And when they do that, that little wheel spins and you produce your ATP. You're doing that in both cell respiration that we talked about earlier, and you're doing it in photosynthesis as well. If you want to get like all fancy, you can look at the orientation of how this happens. And Tyler was alluding to this earlier. Basically, what's happening is it's happening in a different direction in the chloroplast compared to the mitochondria. So in the mitochondrial structure, it's going, you're flowing back into the matrix. That's the very, very inside of the mitochondria. And in chloroplasts, you're going to the stroma, which is like the very outside of the chloroplast. So you're just doing it in opposite directions. It's really the only thing that's different. But it's the same process. I think, chloroplasts and mitochondria generate ATP by chemiosmosis, but they do use different energy sources. Mitochondria is going to use food, right? It's going to use glucose. It's going to use chemical energy. Chloroplasts are going to use light energy. Right? They're using light energy to generate its ATP. Yeah, Grace.
So like how some plant cells have mitochondria, where are they getting the glucose from?
Okay, so that's a great question. So one of the things that I have not talked about yet is the fact that photosynthesis, its whole deal, its whole reason for existence is to make sugar, to make glucose, make a storable form of glucose. Okay, it could be glucose, it could be starch, it could be all sorts of macromolecules, it could be lots of different things. But it's basically making their food, right? How are they going to get the ATP back out of their food once it's there? If I have a glucose molecule, how do I break it down? Cell respiration. So plants perform both, and this is an important point that it's another test question that loves to come up. Plants perform both photosynthesis and cellular respiration. They make their own food, and then they break their own food apart when they need it. Another test question, right? Which one of the following processes happens in plants? Photosynthesis, cell respiration, photosynthesis and cell respiration. I don't know. Neither. It's both. Plants perform both cell respiration and photosynthesis. They make their sugar, and then when they need it, they have to break it apart. They put the ATP into the glucose, and then when they need it, they have to harvest it back out and use it for whatever they need to grow flowers or roots or whatever it happens to be. So this is a summary of that slide that I showed you about the spatial orientation. Mitochondria protons are pumped to the inner membrane space and drive ATP synthesis, and they diffuse back into the matrix. So they're going back into the very, very middle of the mitochondria. In chloroplasts, protons are pumped into the thylakoid space, and they diffuse back into the stroma. So they're going to the very outside of the chloroplast. So the orientation is different. The process is exactly the same, which is what I showed you on this one. Okay. And you can see that difference. Here's the matrix, and here's the stroma. Same process, but in a different direction. Okay. The ATP and the NADPH that were produced on the side. The ACP and the NADPH are produced on the side facing the stroma, which is good. It's beneficial because they're right where they need to be for the Calvin cycle. So Calvin cycle is going to make our sugar. So there's the starting blocks for the production of sugar are right there, right where they need to be. Okay, so again, this is a great kind of diagram that you could spend a lot of time talking through. Do y'all do concept maps? Ever done a concept map? Oh, they used to be so like everyone did concept maps. We might have to do that with you all. Nobody's done a concept map? Who's done a concept map? Somebody's done a concept map. Sure. Okay, that's not. Yeah, we might have to do that. This is a more complicated one that I showed you a little earlier. And actually, this one, I just realized this one, if you wanted to blow this one up, some of the things are not labeled. It's like that isn't labeled. This isn't labeled. Oh, yeah, they sort of are labeled. Never mind. I was thinking you could use that as a tool to kind of help you. But it's still just explaining the process. A lot of times explaining the process really points out, but how does this happen? And that's where you say, I don't know. And that's where you come to me and say, I don't know. How does this happen? And then I can help you with that. So it'll help narrow down where you're confused. Because this is a lot. And I still haven't gotten to the Calvin cycle, which is right there. I just can't do it. I just can't do it today. Yes. You had too much already. Well, we're only 173. Yeah. We have, 13 slides left? Oh, yeah. Calvin cycle's comparatively easier. The crazy thing is there used to be even more in this unit than there was, and they eliminated some of it. Test is not until October 27th, so it's a week from Friday. So we've got time. Yeah, it's not. Oh God, it's not this Friday. No.